Welcome to my channel. Today we are studying about the kidneys, about the kidneys, how it is viewed in the abdomen. We had studied. Then also section of the kidney, also studied. In the section of kidney, outer region is called cortex, and the inner region is called medulla. Cortex and medulla. And after medulla, that means the medulla, in the medulla there are the renal pyramids, and the renal pyramids open into the pelvis. It is a funnel shaped portion. We also say the pelvic cavity. It is the pelvic cavity, the pyramids are protruded. The medulla is ending as pyramids, which are called the renal pyramids. So three portions mainly we study: the outer cortex, inner medulla, and medulla extends as pyramids called renal pyramids, and the renal pyramids open into a cavity called the pelvic cavity or pelvis or pelvic cavity, and the pyramids are opening into the pyramids are protruded into the pelvis, and urine falls into the pelvis. Urine trickles down little by little. Urine trickles down little by little into the pelvis. So these are all the macroscopic structure. Let us study what is seen when a small portion is taken from the section, that is from the region where cortex and medulla are there. From cortex and medulla, a thin slice, a thin part is taken and examined under the microscope. Then the parts will be seen as is drawn here on the board. And when a portion of the medulla is taken, we'll be seeing these sort of parts, which are called Bowman's capsule. These capsules are called Bowman's capsule. Saying what is drawn here is the Bowman's capsule. Which region? They are in the cortex. Bowman's capsules will be seen in the cortex. As a continuation of the Bowman's capsule, there will be a tubule starting from here. From the Bowman's capsule starts a tubule. The tubule is called renal tubule. It is the nephron. What is shown here is nephron. We are studying about the ultrafilters. There are 12 lakh ultrafilters in the kidneys. There are 12 lakh, about 12 lakh, that is 1.2 million ultrafilters in the kidneys. So it stands from Bowman's capsule. Then there is a convoluted part, first convoluted part. Then it passes through a long loop called Henle's loop. And then again there is another convoluted part, that is the second convoluted part, which opens into a collecting tubule. Many collecting tubules are open into that. We are studying about the ultrafilters. Each ultrafilter is called a nephron. We are studying about the ultrafilters. Each ultrafilter is called a nephron. The study of kidney is called a nephrology. So this is what is this figure showing? This figure shows the structure of a nephron. Each nephron stands from Bowman's capsule. See, this is a tubule. We are studying the structure of a nephron. Each nephron starts from a capsule called a Bowman's capsule. It has got the first convoluted portion. Then there is a long loop called the Henle's loop. And then there is a second convoluted part. It opens into the collecting tubule. What are nephrons? Nephrons are the functional unit of the kidneys. They are the ultra filters. There are 12 lakh ultra filters. These ultra filters are called nephrons. Each nephron stands from a capsule called Bowman's capsule. After Bowman's capsule, there is the renal tubule. This much is renal tubule. The nephron consists of Bowman's capsule and the renal tubule. This portion is Bowman's capsule. Then there is a long tubule called the renal tubule. And now the part of the tubule. Each tubule has got first convoluted part. Then long loop called Henle's loop, and there is the second convoluted part. That is about nephron. The ultrafilters in the kidney are called nephron. Structure of nephron. The nephron starts in a capsule called a Bowman's capsule, and it follows a tube called the renal tubule. The renal tubule consists of a convoluted part, which is called the first convoluted part. Then there is a long loop known as the Henle's loop, and there is a second convoluted part which is in collecting tubule. And where are these in? Cortex and medulla. Bowman's capsule is in the cortex. First convoluted part is in the cortex. Second convoluted part also is in the cortex. Then Henle's loop goes down, extends as far as up to the medulla. The Henle's loop is very long and it reaches the medulla. The other parts are in the cortex. Henle's loop around reaches the medulla. And Henle's loop is a very long loop. It is very long. It reaches up to the medulla. And then there is the second convoluted part. After that, it opens into the collecting tubule. Many collecting tubules are opened. Many renal tubules open into the collecting tubule. Many renal tubules open into the collecting tubule. Collecting tubules opening the parts can be seen here. Collecting tubules are long tubules which will open. Many collecting tubules open into the medullary pyramids. Medulla extends as pelvis into the pelvic cavity. Medulla extends as pyramids into the pelvis, and these collecting tubules will open into the pelvis. Many collecting tubules are there. To the collecting tubule, many renal tubules are reaching, and the collecting tubules open into the pelvis, and urine will be trickled down there. And here, this Bowman's capsule we are studying now. It is in the Bowman's capsule the ultrafiltration takes place. This capsule is shown enlarged here. This is called a Bowman's capsule, and this totally called a Malpighian capsule. The term, what is the difference between Bowman's capsule and Malpighian capsule? This capsule alone is called a Bowman's capsule. There is a capsule. Capsule alone is called the Bowman's capsule, and inside there is a network of capillaries. The network of capillaries is called glomerulus, and the Bowman's capsule together with the glomerulus inside is called a Malpighian capsule. The Bowman capsule together with the glomerulus is called Malpighian capsule. So this figure is of Malpighian capsule, and this is also Malpighian capsule. The capsule alone is called the Bowman's capsule, and inside the Bowman's capsule there is network of capillaries called glomerulus. Inside the Bowman's capsule there is a network of capillaries called glomerulus. The Bowman's capsule together with the glomerulus is called the Malpighian capsule. This is figure of Malpighian capsule. So each renal tubule starts from a Malpighian capsule, and this is Malpighian capsule. A branch of the renal artery enters, that is the afferent vessel. This is afferent vessel, the entering one. It enters and this goes out. The going one is called a efferent vessel. The entering one is called afferent vessel. The afferent vessel shown here is a branch of the renal artery. Renal artery divides and subdivides, and the renal artery divides and subdivides, and a fine branch enters into the Bowman's capsule. That fine branch is called the afferent vessel. There is a fine branch entering, that is called afferent, A-F-F-E-R-E-N-T, afferent vessel. And an afferent vessel enters, and it branches inside, divides into many branches. So a network of capillaries is formed here. Then that takes exit from that outside, goes outside. That takes exit from the Bowman's capsule. Then when it makes exit means like going out. The entrance opposite exit. When it makes exit, that vessel is called afferent vessel. That a fine artery that leaves that is called the afferent vessel. Efferent. Exiting is called efferent, and entering is called afferent. An afferent vessel enters, divides into capillaries, and takes exit as an efferent vessel. The entering one is called afferent vessel. That leaving one is called efferent vessel. So these are capillaries, fine capillaries. There is a network of capillaries inside the Bowman's capsule that is called the glomerulus. That is the actual filter. The glomerulus is the, and the one of the glomerulus is very thin. One, that is what is it? Very narrow artery. The wall of the glomerulus is very thin artery. The wall of the glomerulus is very thin. Just one layer thickness is there. The wall of the glomerulus is very layer thickness is there. The wall of the glomerulus is very thin. That is just one layer thickness of cells is there. And when blood is passed through here, what happens? Through here, substances go out of it. When it passes through here, when it passes through the tubule, in the blood there are substances, water, plasma, water and plasma, and certain substances will go, water and plasma, and certain substances will go out. Blood consists of corpuscles and plasma. From the plasma, water will go out. Protein also will go out. Mainly two types of proteins are there: globulin and albumin. Albumin can go out, but globulin big molecules cannot go out. And glucose go out in water. Salts also will go out. Glucose and salts will go out of. And it is like this, and it will go out as filtrate. Glucose and salt will go out, and it will go out as filtrate. And that filtrate is going out through this. When the filtrate goes out through the renal tubule has the capacity to reabsorb substances which are gone out will be reabsorbed. How can the reabsorption take place? This an artery again the efferent artery that leaves from the Bowman capsule gives so many branches to the renal tubule. To the surrounding the renal tubule, this is renal tubule from here. First convoluted part, mainly super, second convoluted tube, all these parts consist of the renal tubule. And to the wall of the renal tubule, the capillary supply is there. The afferent vessel supplies to there. The afferent vessel supplies to there, and that is meant for reabsorption from filtrate that goes out of the filter is called filtrate. When filtrate goes out through here, the filtrate from the filtrate substances are reabsorbed because water should not be sent out. Water is reabsorbed. More water is reabsorbed, and glucose is also absorbed. When it passes through here, glucose will go out into this. Glucose will go out into the renal tubule. When glucose goes out, the concentration of the solution, glucose solution, or the filtrate will be higher, and the density is higher. So osmosis takes place. So from this, when things has gone out, water and glucose and other salts filtered and gone out, the concentration of the filtrate is more. The density of the concentration of the filtrate will be comparatively more than that in the blood. Concentration of the filtrate will be comparatively more than that in the blood. So what happens? Water will be absorbed. That water will enter as osmosis due to osmosis. Water will go out as osmosis. When glucose and salts have left there, the concentration of the filtrate that is passing through there into the filtrate, osmosis will take place. So through osmosis, much water will go. Already water will go. Water escapes through the thin layer. When water has escaped through the thin layer, what happens? Glucose also goes out. As more and more glucose goes out and more and more mineral salts go out, mineral salts go out as ions, sodium ions, potassium ions, etc. will be left. Salts and the salts will be existing as ions. If it is sodium chloride, when dissolved in water, it will be existing as Na positive ions and Cl negative ions. So NaCl, the salt is, but Na won't be seen as NaCl, Na positive ions and Cl negative ions. That's why we can say ions will go out. And potassium chloride goes means potassium ions and chloride ions, like that. Magnesium ion goes, magnesium sulfate goes means magnesium sulfate goes means magnesium ion and sulfate ion, like that. So glucose and ions will have less. The concentration of the filtrate will be more than that of the in the plasma of the blood. So more water will go. As more water goes, it should not take place. And this glucose is a thing required, and this will reabsorb when it passes through the capillaries. So the blood in the capillary blood inside, these will be less only. Concentration of glucose and ions and water will be less inside. So inside the capillary network. So water that goes out required will be reabsorbed. Glucose that goes out will be reabsorbed. Ions that goes out, ions that went out will be reabsorbed. when glucose is more, as in a diabetes, hyperglycemia, more sugar in the blood, more glucose will go out, and more glucose will be uh, absorbed as osmosis. More glucose will go out, and when you get glucose, when more glucose in the blood, reabsorption will take place. But already when there is more glucose in the blood, reabsorption won't take place because blood has got more glucose in it. Glucose that losses here cannot be reabsorbed. And water concentration also. If drunken, we will feel thirst then. One will feel thirst then. When drunken enough water, there won't be thirst because blood will have required homeostasis of having sufficient water. If blood is not having plasma, not having the sufficient water in it, by reabsorption, water will be absorbed. So in short, the required things will be reabsorbed when it passes through the renal tube, and the reabsorption will be according to the requirement. Excess water is there, it will be gone unabsorbed. And excess glucose is there, it will be gone unabsorbed. Excess salt we have taken, that will be gone unabsorbed. So the kidneys filter out things which are not required in the body. So the kidneys filter out things which are not required in the body. So kidneys are the in the kidneys, there are the ultra filters. We say it the nephrons. Nephron is the unit of the kidney, functional unit of the kidney. It consists of ultra filters. Or these nephron ultra filters itself we say the nephron. For ultra filters we say the nephron. And when it passes, the required things will be reabsorbed. So not required things won't be reabsorbed. It helps in the elimination. So kidneys help in the elimination of waste materials from the blood. The blood is purified. When long time excess glucose in the blood, normally 100 milligram per deciliter is required. If much more than that, 200, 300 and that, it will be eliminated through the kidneys. And the glucose will be in the urine. Glucose in urine is called glycosuria. Glycosuria means loss of glucose through urine. And when there is glycosuria, more water also will go to flush out the water. As more glucose goes, more water also will go. So there will be more thirst for the person. And water is done correspondingly. When glucose goes out for a diabetic, when glucose goes out for a diabetic, he drinks more water because he will have thirst, and more water because he will have thirst, and more water urine going is called polyuria. A diabetes patient will excrete more urine. Excretion of urine is called polyuria. And when glucose goes out, he won't be getting, his brain won't be getting sufficient glucose. So he feels that he is starving. Although he has eaten and glucose goes out when there is glycosuria, he is not satisfied in getting eaten. So he will have, although belly is full, he will feel to eat more. That is called brain hunger. It is brain hunger. And you will have more feeling to eat, more appetite, more hunger to eat. That is polydipsia. It will eat more. Polydipsia. And there will be more urine pass. It is called polyuria. If there is glucose in the urine, it is called polyuria. And more thirst is there, it is polydipsia. More water goes out. So he will have more thirst, polydipsia. And the brain is not getting enough glucose and he will eat more. It is for polyphagia. Feeling to eat more is polyphagia. Feeling to drink enough more water, polydipsia, more thirst. Glucose going out, that is glycosuria. Glycosuria. So those are all for diabetes. And for normal person, blood sugar will be nearly 100 milligram per deciliter, and water he will be drunken will be water he drinks will be normal quantity of water. So the kidneys are the ultra filters. When we have taken so much medicines and all excess medicines, so much medicines taking, those medicines and all will be neutralized or changed into some other substance, or as such will be sent out through urine. So kidneys are the filters. The working unit of the kidney is called nephron. That means the ultra filters are called nephrons. Ultra filters are called nephrons. And this renal tubule ends in collecting tubule. Collecting tubule. Many renal tubules are entering into collecting tubule. And there is another figure also given here. That is ultrafiltration. Explaining more about ultrafiltration. About formation of urine. Urine is formed through process like ultrafiltration. The ultrafiltration is to produce urine. Urine is formed through processes like ultrafiltration, reabsorption. First, ultrafiltration takes place. After that, reabsorption takes place. And secretion and absorption of water. Secretion of water means water goes out. From the blood, water is secreted, means water is separated. Water is secreted means from the blood, water is secreted into the tubule, renal tubule. And then water is reabsorbed. So the urine formation, how it is? So process like ultrafiltration and reabsorption and secretion and absorption of water. Urine is secreted. Urine is absorbed also, where in the renal tubule, water is reabsorbed. Analyze illustration 5.5 on the indicators and prepare notes on the notes in the science theory on the process of formation of urine. Urine ultrafiltration. Once more here given about ultrafiltration. When blood flows through the glomerulus. What is glomerulus? The capillary network inside the Bowman's capsule is called the glomerulus. When blood flows through the glomerulus, ultrafiltration takes place through its small pores. Small pores of what? The glomerulus has got small pores. What are glomerulus? The capillary network inside the Bowman's capsule is called the glomerulus, and only a single layer of cells. So what happens? There is gap between the cells. Cells will give part separate for this to pass out. Just like when we are in a queue, long queue, certain persons have to cross and go. Please give side. So a person will move forward, another person backward. A small gap is got through that we will be able to go. Although we can cut a queue and go. Like that, glucose, water, etc. will make a cut through the gap, and the gap is filled. So the cells will give a gap and they will escape. Water will escape and glucose will escape through a small pore. The cells will give way. Temporary pore is produced. This process is supported by the high pressure developed inside the glomerulus. For that, what is required? In the glomerulus, high pressure is to be developed. Blood is flowing slowly without pressure. It won't go out. Suppose we are in a queue. Persons in front of us, a man cuts and go. Or just he will tell us to move backward to the front one, move forward, he will go. If only rush is there, rather no rush means people won't allow, they will be in the queue itself. When there is pressure, it will go, will be. So, blood should flow through this renal tube, glomerulus, with some pressure. No pressure, ultrafiltration won't take place. Kidneys need a definite pressure. In less pressure, it cannot be done. And more, what about more pressure? More pressure means when blood passes through the brain, it will cause for stroke and high pressure. So BP good or bad? More BP is bad. Normal BP is required. Less BP means kidneys cannot work. A certain level of BP is required. But another thing, there is an adjacent kidney region to rise the pressure. There is local pressure increasing. There is a way the body has got. The local pressure will be increased and given. So a definite pressure is required. Very low pressure, kidney cannot work. So ultrafiltration takes place through the small pores. This process is supported by the high pressure developed in the glomerulus. So purposely in the body, high pressure is developed in the glomerulus. Suppose our outside BP is 120/80, inside the kidney it will be given more than that. So local pressure is developed purposely. High pressure developed in the glomerulus due to the difference in the diameter of the afferent vessel and afferent vessel. How is this high pressure developed in the kidney? Due to the diameter of the afferent vessel and afferent vessel. Afferent vessel has got a more diameter, so more blood will enter. But afferent vessel has got less diameter, so blood cannot go out. Will be allowed to enter in, but going out will not be. If into a hall, some queue and more people are allowed into, less people only going out, then it's cannot. In bus itself, more people entering, but less people going out, their pressure will develop. Like that here, afferent vessel is with more diameter. Efferent vessel is with less diameters. That is the technique by which high pressure is developed inside the kidney. Diameters of the afferent vessel is more, efferent vessel less. The glomerular filtrate formed as a result of this is collected in the capsular space. Capsular space means Bowman's capsule space. You are telling about the filtration in the Bowman's capsule. That is in the Malpighian capsule. Bowman's capsule together with the glomerulus is called Malpighian capsule. The glomerular filtrate falls into the Bowman's capsule. At the Bowman's capsule, it will flow to the renal tubule. Components of the glomerular filtrate. What is filtered into the capsule? What are the components? What are the components of the glomerular filtrate? Water. The main component is water. Then what? Glucose. Then what? Amino acids. What are amino acids? Proteins simplest form. Amino acids. Proteins are made into smaller molecules. They are amino acids. Proteins as amino acids. As albumin and globulin, they existed. Of which the albumin smaller molecule compared to globulin. Globulin won't fall out through the glomerulus. But albumin can come out as glomerular filtrate. And it will be reabsorbed. Water. Urine consists of mostly water. Everybody knows. Urine is water. We don't understand that things are there. Just water we feel. But there is some glucose will be reabsorbed. And amino acids. They also will be reabsorbed. But in some diseases, these things won't get reabsorbed. Then what? Sodium, potassium. Sodium chloride. Potassium chloride. Etc. exists as ions. As sodium ions. Potassium ions. Etc. Sodium, potassium, calcium ions. What calcium ions? Calcium chloride, calcium sulfate, etc. Amino acids, sodium, potassium, calcium ions. Then there will be vitamins. There will be vitamins also. For example, B complex vitamins will dissolve in water. It is water soluble, so it can go out. Vitamins, excess B complex is there, it will go out. Then iron salts, excess iron has taken, it will go out. Then urea, uric acid and creatinine. Urea is to be excreted out. Then some uric acid also is there, and creatinine. When the kidney, it is normally are secretion absorbed